Hello, my name is Dr. Young-Anichka. I'm a associate professor at Duquesne University. And I'm going to be producing a little video right now that's going to show you how to access a protein sequence on GenBank, how to get the file, and then how to do a global pairwise alignment using a tool that's available through the EBI server. And then, we're going to use that sequence to make some inferences about the conservation of this particular protein, what are the most likely functional areas that are more conserved, and then I'll show you some other bits and pieces of information that you can access through NCBI. That is, for example, the structure of the protein, how that relates to conservation, and then we're going to discuss some other information that is available on the gene, in the gene database of NCBI, and we're specifically going to focus on expression patterns, and then the gene interactions and phenotypes. And I want to highlight how even a gene that is really well studied and it's been studied for a long time, there's potentially lots of other information that we really don't know much about as far as how this gene is working and how basically, the actions that genes do are simple. They do a lot of different things in different contexts. And so even a gene, in this particular example, it's going to be PAC6, which is a homeotic gene that starts the cascade of differentiation of the eye tissue during development. Even this gene that's a transcription factor in this early development process, in adult tissues, it actually has other functions that aren't necessarily associated with what it's actually been known for and what's been probably most studied as. Okay, so we're first going to grab 3 PAC6 sequences from the human, the mouse, and the Drosophila, the fly. And these are all going to be homologs of each other, right? And so the best place to make sure that the genes that you're grabbing from different organisms are homologous is if you go to the homologene database at NCBI. So I'm going to type PAC6. It's going to take us to the page for this particular homologene record. Now on the left here we have the list of genes that are in the homogenes entry for this particular protein, PAC6, and then to the right is going to be the protein sequence that matches the particular organism on the left, right? And so what we're going to want to do is grab the FASTA file by first clicking on the protein accession. When you go to the protein, we're going to then click FASTA, okay? And we're going to copy this FASTA formatted sequence. So from the greater than sign with the line that has the sequence identifier information to the end of the sequence, we're going to copy and we're going to open up a text editor. So I'm just going to use Notepad. And we're going to paste it into that text file, right? And so we're going to save it right now as just simply, see, I'm going to put it into my folder for this video. And I just call it pac6.txt. So typically when you get sequences from GenBank or other sources, you want to get them as a FASTA file, okay? And then we're going to go back to that homologene record, and then we're going to also click on the mouse sequence, musculus. Again, we're going to click on the protein accession number, then the FASTA link, then we're copy paste this sequence into that FASTA text file. So a FASTA file is a file that has the sequences formatted in a specific way. It's saved as a flat, simple text file. Okay. Next thing we're going to do is we're going to grab the fly one. When the homolog for PAC6 in the fly is actually called twin of eyeless, it's toy for short. So we're going to click on that record. Again, click on the FASTA version of that record. Copy, paste it. Okay. So now we have our 3 protein sequences for the homolog of PAC6 in human and the homolog in mouse and also in fly, okay? And we want to see how similar or different they are, right? So first thing we're going to do is we're going to align using a global pairwise alignment algorithm, the human version and the mouse version, right? So to do that, we're going to go to the EBI, EMBL EBI server page that has different pairwise sequence alignment tools. We're going to pick the global alignment tool needle, and then we're going to paste in our two sequences. So again, the human PAC6, and then we're going to paste in the mouse PAC6. Okay. Then we're going to hit submit. It shouldn't take too long to run, only maybe, 5 or 10 seconds. Now notice when we get this alignment, we have a page that opens up the results. So basically what the server did is it passed the data you input to the program that's on the server, and then it sent the output file to an HTML document, right? And so in the beginning, we have information on the program and the settings that were run on the server when you submitted the job. And then in the second block, we have the results, right? So it says you align two sequences. Here are the session numbers for the two sequences you align. It did a pairwise alignment using the eBlossom 62 matrix. There's a certain gap penalty, extension penalty. But the key thing we want to focus on is look at this perfect alignment. So here's basically the first line of the sequence is the human sequence 1 through 50, and the line below is the mouse sequence 1 through 50, right? And it's basically showing you the alignment for 50 base pairs at a time all the way to the end, right? So the length of both sequences is 436 amino acids, okay? If the amino acid is identical between the two sequences at that position, there's a solid line. If it's similar, the amino acid is similar but different, but it has similar physiochemical properties, there's two dots connecting it. So this Q and this E, it represents amino acids that have similar physiochemical properties, right? So if you look at the sequence identity, we notice that it's 99.8% between mouse and human, right? This actually suggests that this protein is highly conserved. It means that it has some very important function. And of course it is, because it starts to cascade for the development of the eye, right? And if there's any mutations that disrupt this development cascade, then it's going to be detrimental. In a lot of cases, it's going to be lethal, right? So there's strong selection to maintain the amino acid sequence across very diverse lineages, such as mouse and human. Now, the sequence similarity, notice, is 100%. So again, the similarity takes into account the physiochemical properties of amino acids, right? So there's one amino acid difference between the human and mouse, but because those amino acid differences are subtle, and they actually represent amino acids with side chains that have similar physiochemical properties, again, the similarity is still 100%. Now let's look at the alignment between the mouse and the fly, right? So if we align these two, so now we're going to paste in the sequence for the mouse. Next, we're going to paste in the sequence for the fly. And we're going to use the default settings, hit submit, right? Now notice that the sequence similarity is much lower and the sequence identity is much lower. So there's a sequence amino acid identity of only 47.5% between the mouse PAC6 and the toy fly homolog. The sequence similarity is a little bit higher, it's 55.2%. And this suggests that, again, some of those changes are have similar physiochemical properties. Now, if we look at the actual alignment itself, we'll notice that there's actually quite a lot of gaps. Like these parts here, there's insertions or deletions in these different sequences. And for example, the fly one has a pretty long stretch of almost 25 amino acids at the beginning. It has a gap around 60 amino acid position, 60. But there's basically stretches still where there's areas of really high conservation. So for example, from about the second, again, dot represents sequence or amino acid similarity. So from about position 2 in the mouse all the way to maybe about 172 in the mouse, this structure and also the amino acid sequence is actually quite similar. Of course, the actual mouse has a pretty long amino acid insertion in the middle of that. There's another region of pretty high sequence similarity around position, maybe about 207 in the mouse to about 291 in the mouse, right? So there's another stretch that's highly similar, and the rest of them, the rest of that protein doesn't really align. Now, one thing to keep in mind is that the proteins are actually so similar structurally and functionally that researchers that have created knockouts where the fly is missing the eyeless homeotic gene, and because of this, the fly doesn't actually develop an eye. When they've taken the mouse protein and inserted that into the fly, it actually was able to stimulate eye development, right? So basically, even though the mouse protein version of homolog PAC6 is sequence-wise substantially different from the actual fly homolog, there's enough similarity in those really important areas for its function as a homeotic gene that basically they're almost replaceable, right? Again, the mouse version can stimulate eye differentiation in the fly, which is quite remarkable, right? And this again shows that you can have conserved structure and function of proteins that are really functionally important, even though you can have pretty substantial sequence divergence. Now, one important question is, okay, are these areas of high conservation, do they correspond to some important parts of the particular protein? Well, let's see. So we can actually look at the protein structural model. If we go to structures and we search PAC6, we can see what that protein looks like, right? We can compare it to the conserved parts of the sequence, right? Now, PAC6 doesn't have a structure for the mouse version, but there's a structure for the human version. Right, so that's the crystal structure right here. Now, I'm going to click on the full feature 3D viewer. Now, of course, we can use the human structure as a surrogate for the mouse structure because, again, the human sequence and the mouse sequence is 99.8% identical, and it has 100% sequence similarity. So now what do we see here? Well, we see that there's actually these two domains pictured here that are binding to DNA, and you can see that one of these helices is actually fitting perfectly into a major group, right, of a DNA molecule, right? So this again suggests that this is a transcription factor, which it is, and it's showing you that this particular domain is actually fitting into a major groove of a DNA molecule in the area where there would be a regulatory element. So remember, regulatory transcription factors bind to sequences that are regulatory elements, right? And so it makes sense that basically a homeotic gene is a transcription factor, right? Because a transcription factor turns on other genes. The homeotic gene starts a cascade in early development that then turns on a slew of other genes that then turn on other genes that lead to differentiation of tissue, and in this case, into an eye, right? So you can see there's another domain here in this structure that also has an area that binds to, it fits in again to a major groove that is, a little bit farther down the DNA molecule, right? And then you have also these lines that are connecting them. That's, these are basically chains of amino acids that are connecting these two domains that are actually fitting into the minor groove of that molecule, right? So most likely, basically this entire area is the regulatory element sequence that basically binds to that portion of the DNA binding portion of the PAC6 protein, right? Now, if we look at the protein sequence that is represented as structure, we'll notice it's down here, it's right here to the right, and we can even click on it, right, and it's going to highlight it. We actually have only a small part of the entire protein in this particular structure. That's one of the things that you'll find is that for a lot of proteins, the structure that's actually resolved only represents a portion or maybe some of the domains of a larger protein, right? Protein molecule. In this particular case, this DNA binding region, is actually in the very beginning. It's like the first about 150 amino acids of the PAX6 protein. So if we look at that alignment, right, that DNA binding region is about maybe, again, the first 130 to 150 amino acids in the mouse protein, right? And you can see it's actually highly conserved with the fly, so it's one of the conserved regions with the fly homolog, and that suggests, and it kind of explains why the mouse version of the protein can actually function similarly to the fly version and can replace it and start that I differentiation cascade. And it's because the DNA binding portion is similar enough that it binds to the same regulatory elements, right? Now, what do you think this particular portion is that's concerned here, right? It's not in the structural model, but remember, regulatory elements have basically like two components, right? They have the DNA binding domain or domains, and then they have the domains that interact with the general transcription factors or the RNA polymerase complex, right? Because they basically interact with those to recruit the RNA transcription complex and to recruit those general transcription factors, right? So presumably that really conserved section in the middle, between the PAX6 homolog and the toy homolog and the fly, is most likely a region of the protein that recruits the general transcription factors. And because it's similar enough, again, that mouse version can function equivalently, at least in the that particular part of development, as the fly version did, right? So those two can basically substitute for each other. At least in the fly knockout, the mouse protein was able to fill that role and still start the cascade that leads to the I differentiation, right? I also want to point out a few other things. So if we go to the gene database for PAX6, so this is the human homolog, right? We can get a lot of other information on that particular gene, right? And one of the really interesting things in there is I'm going to click on the gene record here for the Homo sapiens. So this gene page for the gene that's in the record in the database, and again in this case PAX6, it starts out with a summary. It has the official symbol, official name, location on the chromosome. Down here we have a summary of what the gene does, right? And then we have again, information on different transcripts that are produced. If we go over to expression, so if we go to table of contents, the information on this gene is basically organized based on these table of contents. So if we click on expression, all of a sudden we see RNA-seq expression data for this particular gene in a lot of different human tissues. And notice that in adult somatic tissue, this particular gene is actually expressed in the brain, most likely expressed in the brain and also the stomach, right? And so this is, again, it's originally was described as a homeotic gene that's active during development, right? The fact that this gene, a transcription factor, is also expressed in the brain and the stomach in adult individuals and adult tissues suggests that it probably has a role that's, or I should say it has additional roles to what we traditionally think of it as just working during development, right? And we may or may not know what these roles are, right? I'm not up on the PAX6 literature, but again, that expression pattern would suggest that it has a lot more roles than simply development. If we go to the back up to the table of contents and we click on interactions, we can see that this particular gene, PAX6, interacts with up to 171 other genes, right? So again, this is partly because it's a transcription factor, so it then regulates a lot of other genes. And again, because it's starting a developmental cascade, this basically affecting a lot of other genes downstream. If we go to phenotypes, we notice that this particular gene, let's see, go up to the table of contents, phenotypes, there's numerous conditions that are caused by mutations in this particular gene, right? And so a lot of them, like coloboma of optic disc, coloboma ocular autosomal dominant, a lot of these are linked to eye development and eyes, and so therefore it makes sense again because one of the important roles that it has is in development of an eye. However, there's also potentially other types of syndromes that it's actually linked to, right? And it's important again to consider that these genes that are highly conserved and function in some important physiological or metabolic pathways, often if you have mutations in them, they're going to potentially produce a disease phenotype, right? And they're more likely to produce a disease phenotype than in other genes that maybe aren't as critically important for something like development or for other really important biological processes. Okay, so I'm going to leave it right there. I hope that you guys found this informative. Just wanted to give you a highlight of where you can access different information, and then how comparing the conservation and similarity of sequences is basically related to, and potentially if you integrate it with the structure and also various other pieces of information like expression patterns, it helps you better understand what this gene is doing, how it's evolving, and how it potentially is affecting and driving phenotypes. Thank you.